Let's take a look at the marker track on a genetic map of wheat. Because this is a genetic map, its coordinates are measured in centimorgans. The marker features on this track are simply named intervals on the map. We can expand the track to see individual marker names, although some of them will not fit on the screen until we zoom in. Let's click one of the markers to open its marker details dialog. As with gene annotations, the Basic Properties tab displays all the common metadata about this marker: its names, marker type, the organism from which this marker was derived, publication source, as well as its location. As always, you can click the map set and the map to view their respective properties. And you can click the eyeball icon next to the marker's location to zoom into it. Some markers may have multiple names. For example, this marker, located on a different genetic map of wheat, has two names: the full name and the alias. The marker's primary name, that is, the name that is shown on the map, is always listed first. The Qualifiers tab at the bottom of the dialog contains a searchable table of qualifiers for this marker, as well as the qualifiers for this marker's location. Unlike a gene annotation, the same marker may be mapped to multiple maps and even to multiple locations on the same map, and the mapping qualifiers describe the properties of each individual marker location. In fact, let's take a look at the All Locations tab, where we can see all the other places this marker is mapped to. In this case, the marker is present in 5 locations, including the current map, and you can click the eyeball icon next to each marker location to open that map and zoom in on the marker. Now let's return to the Marker Details dialog and take a look at the Sequences tab. This tab lists all the known sequences for the marker. In this case, the assay sequences that produced it, as well as the genomic sequence that is covered by the marker on this chromosome of wheat. As usual, you can click this button to copy the sequence to the clipboard, or click this button to align the sequence against other genomes using BLAST-N. By the way, genetic maps are often presented vertically in publications. We can do the same thing by right-clicking anywhere on the gray space between the maps and selecting this option from the context menu.